Assalamualaikum and hello to everyone. Today, my group will explain and present our project about the Saccharomyces cerevisiae. And my part, I will explain to you about the introduction of this species, which is about the Saccharomyces cerevisiae. And firstly, we will listen to the description of the Saccharomyces cerevisiae. The description for the Saccharomyces cerevisiae: due to its distinctive physiology and associated critical functions in many food fermentations and other industrial processes, Saccharomyces cerevisiae is the most commonly used yeast in biotechnology globally. And that is the description for the Saccharomyces cerevisiae. And now I will show to you about the structure of this species. What is the organelles in this species? So let me see. In here, we can see a lot of organelles in the Saccharomyces cerevisiae structure. First, we can see secretory vesicles, fat globule, Golgi apparatus, vacuole, cell membrane, cell wall, mitochondrion, endoplasmic reticulum, nucleus, actin cable, and bud. These organelles have their own functions in Saccharomyces cerevisiae.
So next, I'm going to be sharing about the contribution of the species Saccharomyces cerevisiae to the human. Human has been used the budding yeast or the Saccharomyces cerevisiae in biotechnology for thousands of years. Saccharomyces cerevisiae has been an essential component of human civilization because of its extensive use in food and beverages fermentation, in which it has a high commercial significance. These species have a special ability where they can produce sugar in carbon dioxide. These species also can tolerate with ethanol concentration 15%. The Saccharomyces cerevisiae involved in the production of many beverages such as wine, beer, and cider. Saccharomyces cerevisiae is also used in the biofuel production industry. This species is also mainly implicated in many fermentative spoilage of high sugar foods and beverages. This is simply because Saccharomyces cerevisiae can tolerate ethanol concentration up to 15%, it may occasionally spoil alcoholic beverages, including wine and beer, and also isolated from dairy products, including milk, yogurt, and cheese. There are many applications of Saccharomyces cerevisiae that we have been used. For example, the first contribution of the species to the human is in the wine and beer industry. Saccharomyces cerevisiae mainly have been used in fermentation of beer and wine. The yeast used in beer making process ferment the different type of sugar found in the wort and the pre-fermented beer to produce ethanol. These species have been used for thousands of years. It is believed that the beer technology was acquired from Germanic and Celtic tribes. Century. However, the oldest known example of yeast used in fermentation of a beverage was found in China back in 7000 before Christ. The next contribution of the species is in the baking industry. Saccharomyces cerevisiae used in baking industry as a commercial preparation that consisting of dried cell of one or more strain of the fungus Saccharomyces cerevisiae. It is used as the leavening agent in the rising of the dough for baking. A secondary contribution of yeast to bread is flavoring and aroma. Baker's yeast or the Saccharomyces cerevisiae often marketed in two ways, either as compressed cake or a dry powder. By feeding on a sugar from the starch flour, yeast produce carbon dioxide. This gas expands the gluten proteins in the flour and causes the dough to rise. This process of bread making being the most commonly associated with the yeast. The third contribution of the species to human is food spoilage. Food and beverages spoilage by yeast is well documented in the literature. Spoilage is a consequence of yeast growth in the product. Yeast spoilage becomes evident to the consumer in many ways depending on the product. The ecological principles that govern the occurrence and growth of the microorganisms in food in general also apply to yeast spoilage of food. Examples of food that apply this technique is yogurt, processed food products like juices, puree, fruit pieces, lemonade, and many more. Bakery products containing fruit are also susceptible to spoilage by Saccharomyces cerevisiae.
The primary role is to convert the sugars, namely glucose present in the grape mass, into alcohol. Did you guys know that Saccharomyces cerevisiae is the most common yeast in winemaking, and it is used to carry out alcoholic fermentation in wine production? It conducts fermentative metabolism to ethanol and carbon dioxide, which is a primary fermentation metabolite as the cells strive to make energy and regenerate the coenzyme NAD+ under anaerobic condition. In the absence of oxygen, the cell will continue some metabolic functions like glycolysis, but will also rely on other pathways such as reduction of acetaldehyde into ethanol to recharge the coenzyme needed to keep metabolism going. It is through this process of fermentation that ethanol is released by S. cerevisiae as the waste product. The more sugars in the grapes, the higher the potential alcohol level of the wine if the yeast is allowed to carry out fermentation to dryness. Note again that this metabolic pathway, glycolysis and reduction of acetaldehyde, is included in alcoholic fermentation of S. cerevisiae. Besides providing the alcohol content in the wine, S. cerevisiae also have some secondary fermentation metabolites that most of it is to provide sensory or important flavor to the wine. Two of the main secondary fermentation metabolites are glycerol and succinic acid. After ethanol and carbon dioxide, glycerol is the largest concentration metabolite. Some winemakers consider the presence of glycerol in wine to impart mouthfeel as a contributor to the viscosity of the beverage. Succinic acid may be produced by S. cerevisiae following a limited operation of the citric acid cycle, whereas glycerol may be produced in redox balancing reactions or as a compatible solute in response to osmotic stress. On the other hand, esters represent a very important group of flavor-active compounds that generally result in desirable fruity or floral flavors and aromas in fermented beverages. Most of these are produced by yeast during fermentation in reactions between alcohol and acyl-CoA molecules. So remember, what is S. cerevisiae roles in winemaking? It is to give the alcohol content and providing flavor of desired into the wine.
Next, I'm going to explain to all of you about the 5 steps of the winemaking process, which are harvesting, crushing and pressing, fermentation, clarification, and then aging and bottling. Harvesting, sometimes known as plucking, is the first step in the winemaking process. There will be no wine without fruit, and no fruit other than grapes can reliably produce enough sugar to yield enough alcohol to retain the ensuing beverage, nor do other fruits have the necessary acids, esters, and tannins to consistently produce natural stable wine. The grapes must be harvested at a specific time, preferably when physiologically ripe, in order to make great wine. Before crushing the grapes, winemakers sort the grape bunches, filtering out any rotting or under-ripe fruit. The next step in the winemaking process is customarily to crush complete clusters of fresh ripe grapes. Mechanical pressing has increased the quality and lifespan of wine while minimizing the need for preservative on the part of the winemaker. When making white wine, a winemaker will quickly press the mass after crushing to separate the juice from the skins, seeds, and sediments. By doing so, tannins and undesirable color from the grape skin rather than the juice are prevented from leaching into the white wine. Fermentation is the true wizardry at work in the winemaking process. If left to its own devices, must or juice will organically ferment in 6 to 12 hours with the help of wild yeast in the air. Many winemakers, however, choose to interfere at this step by inoculating the natural must for a variety of reasons. This means they will destroy the wild, often unpredictable native yeast and replace them with a yeast strain of their choosing, allowing them to forecast the eventual result more accurately. Fermentation usually continues until all of the sugar has been converted to alcohol and a dry wine has been formed. Fermentation time might range from 10 days to a month or longer. The clarifying process begins once the fermentation has finished. Winemakers can rack or siphon their wines from one tank or barrel to another in the hopes of leaving the pomace, which is the precipitates and sediments at the bottom of the fermenting tank. Filtering and fining can be done at this point as well. The maturing and bottling of wine is the final step in the winemaking process. The winemaker has the option of bottling the wine right away after clarifying. Aging can take place in a bottle, stainless steel or ceramic tanks, enormous wooden ovals, or small barrels known as barriques. So, these are the 5 steps of winemaking process to make the good wine.
As a conclusion, we can conclude that Saccharomyces cerevisiae have evolved with humans for many thousands of years. It is benefit in making food like bread and beer that have become staples for our diet. Today, it has become important in different ways, especially in the production of ethanol and biofuel. Maybe Saccharomyces cerevisiae is the future answer to our fossil fuel problem. This is why we need to continue study this simple eukaryote microorganism.